Alright, in this lecture I'm going to be talking about cell division. So, cells dividing via mitosis or meiosis via asexual or sexual reproduction. So, let's go ahead and get into it. So, let's have a little bit of an intro first. The difference between asexual and sexual reproduction is that asexual, A meaning not, so not the sexual manner, is an organism that basically clones itself. So if you look at the right example, if you have some plant that reproduces asexually, then it will create, so you see this dotted line separates up, these are the parents, down, these are the offspring. So in asexual reproduction, to the right half, the parent, this purple squarish blob, let's say it's just an abstract representation of a plant that reproduces asexually. It will produce offspring that is exactly identical to it because it's not mixing its genes with any other parent. So it's just one thing to another. Or if this is some type of bacterium, one bacterium or multiple bacteria, some types of bacteria or one type of bacterium that is reproducing asexually, all of it is going to be the same. All of its offspring are just going to be clones of itself. Now, sexual reproduction, which is also some plants can reproduce sexually or asexually depending on what type of plants they are. But sexual reproduction, such as in animals and plants and humans, but sometimes in biology we call humans animals, there's obviously a male and a female cooperation. So their genes will mix and produce offspring that are also mixed. And so the offspring in sexual reproduction are different from their parents, but the offspring in asexual reproduction are exactly the same. Every single gene is the same as the parent. Okay. For example, so yeah, just what I went over. So asexual daughter cells. So the way we refer to offspring as well is daughter. We don't ever say sons. I don't know why. So, but daughter cells in mitosis, daughter cells in meiosis, daughter plants. I guess daughters in humans, but daughter organisms, just talking about the offspring generally, we can refer to them as daughters. So a daughter plant for this parent plant, and then we obviously refer to parents as parents, is going to be genetically identical and have the same number of genes as the parent. As opposed to a sexual daughter cell or plant or organism, then that will have genetically mixed variation from its parents. Here we have this ovum or egg from this mother, 23 chromosomes, and sperm from the father, 23 chromosomes. They come together, and 23 plus 23 is 46 chromosomes. It forms one-celled organism called a zygote, and that is where the human cell division first begins. This is where I think, on a personal belief, life begins, but that's just my personal opinion. Anyway, so have the same number of genes as each parent, but half from one and half from the other. Okay, so they still have the same number of genes as the mom, still have the same number of genes as dad, but only because the embryo, the offspring got half from one and half from the other. And same thing here, the daughter plant cell has the same exact number of genes as the parent organism, in this case the parent plant, but it doesn't have half and half from two different parents. It just has all of the same ones, but here it has the same number, but they're mixed. Alright, so let's get into mitosis and meiosis now that we kind of went through that. Mitosis is a process where a cell is divided into two daughter cells. There we go again, basically the offspring cell. So here's the mother cell or the parent cell. And then it gets split up into two daughter cells here with a complete and exact copy of all chromosomes from the original DNA. So you can see here, excuse me, the mother cell has this blue strand right here, and it has this green strand right here, and then it just duplicates them each, and then each of them gets separated, and then the two daughter cells thus have one blue strand each, just like the mother, and one green strand one green strand each, just like the mother. So it has a complete and exact copy of all chromosomes. Now, meiosis, on the other hand, is a cell is where a cell is divided into 4 daughter cells instead of 2, each with a copy of half of the chromosomes from the original DNA, and it's mixed each time. So watch what happens is, you can see here there's in meiosis, there's like 2 cycles of it. It's meiosis 1 and meiosis 2, but after meiosis 2 finishes, then that's the end of meiosis. So after meiosis 1, you can see here, at the very start, you can see here that the original parent cell has 2 blue strands and 2 red strands. It's kind of blurry, so I apologize for that image. Then, some mixing happens in meiosis here, where you can see some red over where the blue strand was and blue over where the red strand was, and that's called crossing over, which we'll get into. But that doesn't happen in mitosis. Mitosis is just like it clones of itself. So you can think of mitosis as like for asexual reproduction, and meiosis is for sexual reproduction. So here then they come together. They this crossover thing, and then they get mixed, and then out of those, then they get split up. So we'll go into this in more detail, obviously, but here's just a little introduction to meiosis. So it ends up with 4, but notice how it doesn't have like one of the strands. So actually, this is not a good. This should really start off with 1 blue and 1 red, but I guess that's before this maybe. And so, in comparison to just 1 blue strand and 1 red strand, this daughter cell has 1 strand because it only has half of the chromosomes. But this 1 strand is mixed. It has a little red tip that came from this crossover that happened over here. This one maybe has like exactly just 1 blue strand. This one has exactly 1 red strand, and this one is almost a complete red strand but a little bit of blue. So meiosis leads to a lot more variation. So sexual reproduction leads to a lot more genetic variation in offspring because of that crossing over. All right, so differences here. Again, mitosis splits up into 2 cells that are exactly the same as the parent cell. Meiosis splits up into 4 cells. You can maybe say half cells, 4 cells with half of the original DNA, and none of the 4 are the same. They're all mixed up. This is just like a simple, what's it called? Drawing of it, but it's much more detailed and complex than that in meiosis. There is this video you can watch on your own time. It's really great at illustrating mitosis. All right, so when does mitosis and meiosis happen? Okay, so they happen in a small portion of the cell's life cycle. So cell division happens in this this is one just for mitosis, but meiosis is the same thing. So if you see these arrows on the outer part of the circle here, mitosis is maybe, I don't know, let's say 10%, 15% of a cell's life cycle, and then cytokinesis, we'll get into, it just basically means mitosis finishing and then 2 cells splitting apart. And then most of the cell's life cycle is in this phase called interphase. It's the inter meaning between phase, just phase. So this between phase between mitosis events, mitotic events or meiotic events. So what happens during this interphase? The cell replicates all of its DNA and the cell's functioning normally. So if it's a skin cell, it's going to be doing skin things. If it's like a scalp cell in your head, it's going to be helping to produce hair. If it's like a liver cell, it's going to be helping doing things like cleaning the blood like the liver does. And so whatever it is, if it's whatever it is, it's going to be doing those functions during interphase, and then it's going to take a break, split, divide itself, and then go back to its functions. So let's go through these. The interphase is split up into 3 smaller phases called the G1 phase, the S phase, and the G2 phase. So G is for growth and S is for synthesis, meaning just to make or to build. So G1 is like the first growth phase, and then it goes into this DNA synthesis phase, and then G2 is the second growth phase. Okay, so in G1 phase, the cell is metabolically active, duplicates its organelles and cytosolic components. That just means that it's just getting ramped up. Like the new cell is getting ramped up, they're growing again, and is doing what it needs to do. Then in the S phase is when DNA is replicated. And this kind of is happening throughout the whole time as well, but this is when it mainly focuses on DNA replication, is during the S phase. And then during the second growth phase, G2 phase, it's creating enzymes and protein synthesis occurs. ATP is produced in large amounts. Centrioles replicate, so it's kind of growing the tools it needs, centrioles and energy, in order to enter mitosis. Right? So throughout this whole process, there are tons of checkpoints. Why are there tons of checkpoints? Because in science, in our body, these cells, all of these things, DNA doesn't know what it's doing. The cell doesn't think. The cell is not thinking. The mitochondria are not thinking. The DNA. None of these molecules know what they're doing because they don't know anything. They can't. They don't have a brain to know. They can't know. And so, how does everything work the way it's supposed to work? Well, the only reason it works the way it's supposed to work is because of chemicals mixing and bouncing each other. That's why the cell is so saturated with proteins and chemicals and organelles, because statistically, if there are so many of these things just like bouncing around, then these processes will happen. So there are checkpoints that usually happen throughout these cycles, and the three most famous checkpoints are these ones. So the first, let's say, coming out of mitosis, here's the new cell right here, entering interphase. So this is like the mitotic or meiotic phase right here. And then you have a new cell here, and then the G1 checkpoint is after the first growth. The cell, so there's a checkpoint for checking the cell size and DNA damage. Because hopefully throughout all that mixing and clashing of DNA and chemical signaling, hopefully nothing got missed. And so there are this chemical checkpoint that checks for cell size and DNA damage, and if there is, then there are proteins that will detect it chemically and be activated in order to repair the damage, or the cell will just be consumed by another cell because it'll shut itself down because it's too small or too damaged. Then it'll enter the S phase during the interphase, which is the DNA synthesis, and then second growth phase, there's the G2 checkpoint. There's a G2 checkpoint at the end, just before it enters mitosis or meiosis, and that's where the cell checks whether DNA has doubled and repairs for it repairs it if damaged. So after this DNA synthesis, it doesn't want to create too many doubles, so it just makes sure that everything's all squared away before it gets split in half, and then during almost close to the end of mitosis, it checks whether something called the microtubules have attached to the centromeres of the chromosome. So basically, it makes sure before it splits in half, before it splits the DNA in half, there's a checkpoint that makes sure, all right, are all the DNA exactly in half, and are these microtubules ready to tear the DNA apart into two halves? And after that checkpoint, then they are ready to split in half and then start again, create two new cells that go through this process. All right, so some side note vocabulary. Chromatin is just, you're going to hear a lot of words starting with C, particularly CH as well. Chromatin is just another word for DNA. You can refer back to this slide anytime you need, and I'll be mentioning it as well. Chromosome is basically condensed chromatin, which is DNA. So you can see here, this is a chromosome. This is the way we usually see DNA, but it's not like this throughout most of the cycle. Most of the cell cycle, it's tangled and it's unwound. Right here, it's wound tight in little coils. It just looks like solid red, but there's just a bunch of string wound up like a DNA string. Chromatin just all wound up in there, and it just looks like this. So this is a chromosome. When it gets duplicated, these two are called sister chromatids. So this is one chromatid, this is another chromatid. And these are two daughter chromosomes after they're split after mitosis. and then there's a little protein called centromere, and it holds the two sister chromatids together until they get split apart. This is where the